And so now we'll transition to Dr. Cubb, who will be talking about treatment for anaplastic thyroid cancer. And so I know we got into it a little bit, but Dr. Cubb, BRAF V600E is the most common mutation or alteration. What other drivers are commonly seen in anaplastic thyroid cancer?
So, Dr. Johnson, perfect segue into what we are going over now. So, as he brought up, we think anaplastic has likely developed from a more differentiated thyroid cancer. So, the slide looks really busy, but basically, what it's getting at is the blue square is papillary thyroid cancer, purple square is follicular thyroid cancer, poorly differentiated thyroid cancer. And then in order to get to the orange square, you see all these other arrows with additional mutations like TP53 mutations, mTOR mutations, etc. So basically, we think this anaplastic thyroid cancer has gained other mutations over time that causes it to be more aggressive. It is important to note, though, that it doesn't lose the initial driver. So it's not surprising that we're still seeing BRAF V600E be present in around 40% of anaplastic thyroid cancer. We believe it's present in over 70% of papillary thyroid cancer. We see anaplastic thyroid cancer be RAS driven in around 25%. And then sometimes we could even see oncogenic fusions, which are more rare, such as RET, NTRK, and ALK fusions. We'll get into why this matters and why we care so much about finding this information in some of the later slides.
Yeah, and I think going back to again what Dr. Johnson started talking of was the molecular profile of an anaplastic thyroid cancer, and that's what he discussed was looking at through the tumor. What other technologies are out there to try to get some of the information about the genomics of a tumor?
Yeah, and I know sometimes it can be kind of odd because, after you've had multiple needles stuck in, why don't we have the answer? As Dr. Johnson's figures showed earlier, these tumors can be big and they can have areas of what we call necrosis, which are basically broken down cells that we can't get this information from. So sometimes, in addition to maybe needing an additional biopsy, like a core biopsy, another thing that we can do is actually run a test on your blood. And we look for DNA that's being shed by the tumor. This doesn't always give us a result, but it can help us get some more information when we're waiting either for biopsies to happen or waiting for the next generation sequencing to be run on tissue. Of note, Dr. Johnson did bring up that we could have the IHC done, which is that test that they're able to do within 24 hours. Regardless, we still recommend getting the full tumor next generation sequencing. Just to put it in perspective, that IHC checks for one gene, whereas the next generation sequencing checks for typically over 500 genes. So it's a lot more information.